Hey guys, welcome back. So you guys wanted another biology paper. We have biology paper 1 on Friday, so hopefully this is going out today, which is Wednesday, and we can kind of go through it and just brush up on all of our knowledge. So this is the 2022 paper, hour and 45 minutes. It should take us less time than that, but I'm going to explain each question and just exactly where the misconceptions are, so that way you can go into the paper on Friday, hopefully armed with the correct knowledge that you need. For the first question, we have Gregor Mendel studied inheritance in pea plants. Pea plants can be either produce yellow pea pods or green pea pods. Mendel crossed the plants that always produced yellow ones with plants that always produced green ones. A is a dominant allele and little a is a recessive allele. So what is the genotype of the pea pods produced from this cross? Well, the yellow or green, one of them is going to be dominant, one of them is going to be recessive. So we don't know what that's going to be, right? But the idea is the parents are either going to be both dominant and both recessive, or you're going to have one dominant, one recessive, and both recessive. The reason for this is, let's say, hypothetically, being green is recessive and being yellow is dominant. So for the plants to always produce green pea pods, they must have both recessive alleles, right? Because it's recessive, so that means you need two copies of the allele in order to present that genetic trait. Whereas yellow being dominant, we can either have both be dominant, both alleles be dominant, or one be dominant and one be recessive, because the dominant one will win out. So, what would the children have? Well, if you think about it, whenever you mix genetics like this, you get one allele from the one parent and you get one allele from the other. Now, if we look at the combinations, if we take one from here and one from here, we're always going to end up with A and A. If we take one from here and one from here, we're either going to get A and A, or we're going to get two A's. So, what is kind of the genotype? Well, it's the one that's going to be the most likely. So it's going to be the A, so the capital A and the lowercase a, because that is the one that is produced most frequently. And again, it doesn't actually matter which way round it is, by the way. If you said that green pea pod, the green colour was dominant and the yellow was recessive, all you would do is swap those two around. It wouldn't make a difference, right? The idea is that the highest probability is that we're going to get a dominant and recessive allele almost all of the time. And this is true with your parents as well. So generally speaking, if one of your parents has a recessive trait and one of them has a dominant trait, most of the time you're going to get the dominant trait inherited in the children. And for the next bit, Mendel grew many plants from the seeds in these pea pods. These plants were then crossed with each other. The seeds from this second cross produced 5,496 with yellow and 1,800 with green. So it does look like green was a recessive trait actually. Give this as a ratio. So if we're doing yellow to green, initially we have 5, 4, 9, 6 to 1, 8, 3, 2. And then all we really need to do is simplify the ratio, which is a bit of maths. I would probably just divide both sides by 1832. It does look like it is divisible actually. So if I divide both sides by 1832, you can use a calculator, of course. You actually get the ratio of 3 to 1. So let me show you that just to prove it. Let me open up my calculator. There we have it. So dividing this left-hand side by 1832, we get 3. So it's a 3 to 1 ratio. There are 3 times as many yellow plants as green plants. Complete the Punnett square to show the outcomes of a cross where both parent pea plants are heterozygous. Now, you need to know what this word means. Now, in basic terms, it just means that you have one of each trait. So that means you have the recessive trait and the dominant trait. So in this case, you're going to have A and lowercase a. Other way round also works. It's not going to make a difference in a Punnett square. So we're going to fill that in now. So the parents, you always put the traits that you have, like so. That. Show the percentage probability of the homozygous recessive. So homozygous means you have only one trait. So only one allele, if you like. And it can either be the dominant or it can be the recessive. In this case, we're looking at the recessive trait. So essentially, we're working out the probability that a offspring will have lowercase a in the answer, because that's homozygous because it's both the same allele, and it's recessive because it's the lowercase letter. Remember, capital letters are dominant, lowercase letters are recessive. Then all you do is you match it up. AA. Capital A, lowercase a. Lowercase a, capital A. Lowercase a. Just like so. So, in this, we have two homozygous offspring. One is for dominant, one is for recessive. And we have two heterozygous, because it has one of each trait here. So the probability of having homozygous recessive is 1 in 4, which I'm hoping you guys know from maths, but if you're not too sure, all you need to do is take that fraction and times it by 100 to get 25%, and that's your answer. So if I were to ask you, what's the probability of getting, the probability of getting a homozygous dominant offspring, it's still 1 in 4, so 25%. If I asked you the probability of getting a heterozygous offspring, that's 2 out of 4, so that's a half, which is 50%. So, I hope that makes a lot of sense now. For part C, part 1, it says some plants reproduce sexually. Give one advantage of this type of reproduction. So why is this a good thing? Well, generally speaking, it allows for genetic variation. So it allows genetic variation in offspring. Now that's all you need to write for your mark. However, why is this good? Well, the idea behind it is it does allow for evolution and natural selection. So if all human beings were clones of each other, right? We all reproduced asexually, we would all be identical to each other. Which means the second a disease comes around, we'll all die. The second there's a selection pressure, we all die. And that's the big issue with things like viruses and bacteria, right? Is that we can take a medicine and kill basically all of the viruses or bacteria in our body, within reason anyway. However, when you have genetic variation, you are far more likely to have individuals survive a new selection pressure, whether it be a disease, a change in environment, whatever it may be. So this right here, genetic variation, allows for evolution, natural selection, all of those things as well. But just saying allows genetic variation in offspring gets you the mark. And then lastly, name the process that forms gametes for sexual reproduction. So what they mean is, what type of cell division is this? Is it mitosis or meiosis? Meiosis is the answer, because it produces genetically distinct gametes, whereas mitosis always produces identical clones. So keep that in mind. Mitosis is identical, meiosis is allows for genetic variation. Here's probably one of the best questions you can get in the exam. DNA molecules contain base pairs. Remember, this is things like adenine and thymine or guanine, etc. So you have those A-T pairs or C-G pairs, etc. And it says, describe how these base pairs are bonded, and you get 2 marks. You can actually write quite a few different things. So the first thing you can say is, and this actually gives you 2 marks, is you could literally just say weak hydrogen bonds. That's 2 marks. How cool is that? Just saying weak hydrogen bonds, even if it was on one line, so even if you just moved this up here, that gives you 2 marks, which is so good, because it's 3 words. That's almost a mark a word, pretty good. You can also say H bonds, by the way, which is interesting. The other thing you could say is you could say they're bonded with, so these are kind of additional points you can make, complementary bases. So the idea that every single base has only one other base that it will bond with. And then you can also give an example. So you can either say like A bonds with T, you can say C bonds with G, for example, something along those lines. But that gets you the 4 marks, so you don't really need to worry too much about it. With this, this is just a bit of memory, unfortunately. So figure 1 shows part of a DNA molecule. You need to write the complementary DNA strand, not stand. So what bonds with T? Well, it's always A with T, so we can fill that in right now. I'd fill that in right now, like all of them. Every T I see, I'm going to write an A. The reverse is true. If T and A always bonds, then with T and A, that means every A is going to be a T, so I'm going to fill that in. The only thing we have left now in our strand is G and C. Okay, so that means every G must bond with a C and every C must bond with a G. That's how I do it. So I wouldn't necessarily memorize all of them. I would just kind of maybe remember one of them. So if you remember that A and T bond, then anytime you see a T, you're going to write A. Anytime you see an A, you're going to write T. And then the only two letters you have left are G and C, so they must bond together as well. So anytime you see a G, you write a C. Anytime you write a C, you can write a G. So you can kind of do a little bit of a cheat code like that. Make it a bit easier for yourself. So just remember one pairing, and the other pairing will be just process of elimination, basically. So here it says 3 bases code for each amino acid. It's always the maximum number of amino acids coded for by this strand of DNA. And this is the strand they're talking about, by the way. Or the second one, whatever. So all you need to really do is, if I times all of these numbers by 3, that will give me the number of bases required for that number of amino acids. So it says 3 bases for each amino acid. So if there's 3 amino acids, times it by 3, I would need 9 bases. Now, how many bases do I have here? Well, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So I'm looking for whatever one gives me 12 bases. But there's actually, in my opinion, a slightly easier way to do it. What I could do is just break these off into proteins, right? Into amino acids. 1, 2, 3, 4. So I know the answer must be 4. Again, if you want to go through this, you can times it by 3 and have a look. So here we'd need 12 bases, and if you notice we do have 12 bases, that's the only one that could work. 6 times 3, I would need 18 bases, so I haven't got enough. 12 times 3, I would need 36 bases, which means I don't have enough again. But I actually kind of like the first method, grouping them together, so there's amino acid 1, 2, 3, and 4, so the answer must be 4, because that's the only... So for example, with the first one, I have 9 bases, but we're looking for the maximum number of amino acids. I can still fit another amino acid on the end. So there's a couple of ways you can think about it. For part 3, what is the shape of a DNA molecule? It is a double helix. Fairly famous shape. Part C. A student wanted to extract the DNA from fresh peas, as you do. The student crushed the peas and added washing up liquid and water, but then also added the enzyme protease, which was added to this mixture. Explain why the enzyme protease was added to the mixture. Now, in my opinion, if I wasn't sure about this question, I would think about what protease does. Protease is an enzyme that breaks down protein. I'll write proteins into amino acids, right? Into amino acids. And I'm thinking, okay, well, I know that DNA actually is made up with proteins. Okay, that's one thing. But what else is made up of proteins? Well, enzymes. So I'm thinking, if I'm trying to expose this DNA, I really don't want it to be damaged by anything. The problem is, when we crush up these peas and add them to washing up liquid and water, there are enzymes that exist inside the peas that might break down the DNA itself, which is bad. So we are going to add protease, because protease will break down proteins. Enzymes are just folded proteins. So the protease will gobble up or break down the enzymes that might damage the DNA. That's the point of adding protease. I'm not going to lie, that's a bit of a leap from this to, oh yeah, we need to protect the DNA. But what I'm trying to get you guys to do is think about, what does protease do and what's the goal? I want to be able to see DNA. So the protease must somehow allow me to see DNA, and it allows that by preventing the destruction of the DNA. I hope that makes sense. Again, this is a bit of a memory task as well, but hopefully when it makes sense, it will help you remember. So protease will break down enzymes in the cell membrane that destroy DNA. So in terms of where do you get the marks here, that's a W, not an N, but okay. Where do you get the marks in this question? Well, saying what protease does, so protease breaks down proteins or enzymes, will give you 1 mark. Saying where this happens in the cell membrane will give you another mark. And then that destroyed DNA will give you the third mark, but obviously this is a 2-mark question, so you don't need to worry about that. For part 2, it says the mixture was then heated and filtered. Finally, the student poured the filtrate into a test tube, and ice-cold ethanol was poured down the side of the test tube into the filtrate. Why did they use ethanol? Well, the reason is DNA is actually insoluble in ethanol, which means you'll be able to see bits of DNA floating around in the ethanol because it hasn't dissolved. That's the reason. So DNA is insoluble in ethanol. So why is that important? Again, you don't need to say this, but you can if you wanted to get a mark. Is because it's insoluble. I mean, a good way to think about this is if you know when you pour loads of salt into water and eventually the salt doesn't dissolve anymore, it becomes a precipitate. You can see the salt, right? You can see the salt floating around in it. Same thing here. If we left it in water or by itself, we can't actually see the DNA. However, because it's insoluble in ethanol, you will see strands of DNA floating around in the ethanol, which will help you because, I mean, what's the whole point? He wants to extract it, he wants to be able to see it, right? That's the whole point of the experiment. This is basically maths. So figure 2 shows the number of people diagnosed with STIs in the UK during 2017. So chlamydia, gonorrhea, blah, 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 blah. State the STI that has the median number of people diagnosed. And if you remember how median works from maths, you need to put the numbers in order and then cross them off from each side. So I'm going to put these STIs in order from smallest to largest. So instead of writing out the names because I'm lazy, I'm going to give them letters. So we have syphilis. I'm going to call this GW. I'm going to call this GH. I'm going to call this just G. I'm going to call this C. So if I put it in order from smallest to largest, syphilis is the lowest. Then we have GH. So I'm actually going to tick these off as I go along. Then we have G. Then we have GW. Then we have C. So that's in order from smallest to largest. Now all you do is you find the middle one. And you can probably see that the middle one is G. But just to be super clear, you can cross one off each side. And G is the one in the middle. And G, again, you wouldn't just write G. You'd have to write gonorrhea. So G-O-N, thank God, the name of the disease is there, because I would have forgotten. Anyways, the population of the UK in 2017 was 66 million people. Calculate the total number of people diagnosed with chlamydia in 2017. Well, here it says, number of people diagnosed per 1,000 of the population. So, there are 3.7 every 1,000. What I'm going to do is I'm going to work out how many thousands of people there are in the UK. So we have 66 million divided by 1,000 will give me how many thousands of people. So 66 million is 66 followed by 6 zeros. Over 1,000. You can use a calculator or you can just cross off the last 3 zeros. So there are 66,000 thousands of people. So we're going to do 3.7 multiplied by 66,000. It's pretty handy, right? And that will give us our answer. I don't know how to, could I do that in my head? Maybe, but yeah. I don't want to be arrogant, you know. I'm going to use a calculator. 244,200. I promise I can read numbers. Bad. Now, here's an interesting one. Well, there's two interesting ones. State why chlamydia can be described as a communicable disease. So what is communicable? Communicable means that it can be spread through contact between people. And in fact, that's actually the 1 mark answer you can give. So it is spread through person-to-person contact. Person-person contact. That is one very valid way of saying it. That's why it's communicable. The other thing you could say is that it's caused by viruses, bacteria, protists, etc. In this case, caused by bacteria, because it's not a viral infection. But I'm going to forgive you if you don't remember that chlamydia is a bacterial infection. So I would probably just stick to this one. But if you happen to know if it's a virus or bacteria or whatever, you can say that because all bacterial infections are communicable because you can catch them from other people. So communicable means you can catch it from someone else. So something like the flu, the common cold, etc. A non-communicable disease is something that you, generally speaking, inherit from your parents or you develop over time. So something like diabetes, something like cystic fibrosis, or some kind of genetic condition, because you can't spread that to another person. Give one way the transmission of chlamydia can be prevented. Well, it's a sexually transmitted infection, so you can just say avoid sexual contact. You can also say, using a condom, not just contraception. Remember that, it has to be a condom. Screening people for infection, treating it with antibiotics. So for example, if one person has chlamydia, if you treat them, then obviously they won't spread chlamydia to other people. Things along those lines. Explain why chlamydia can be treated with antibiotics. So if you forgot that it's a bacterial infection, you're kind of screwed now, because antibiotics only work with bacterial infections, which is a really common misconception. You can't have it for something like the common cold or flu. It has to be a bacterial infection. So, because it is killed by antibiotics. Because it's a bacterial infection. Simple as. HIV is another sexually transmitted infection. Explain how HIV can lead to the onset of AIDS. And again, this is another common misconception that HIV and AIDS are the same thing, or that AIDS is actually the disease. AIDS is not a disease, it's a symptom. So HIV is human immunodeficiency virus. It is the virus that may lead to AIDS and generally does. But AIDS is assisted immunodeficiency syndrome. Basically, your immune system stops being able to fight any kind of infection, even the ones that we're exposed to all the time. So a lot of people say, "Oh, the common cold would be very bad for an AIDS patient." It's even more brutal than that. Every day you're exposed to bacteria and viruses. Every day. And your immune system is strong enough that you don't even ever get sick. So there are diseases that we haven't named because our bodies take care of them and no one's really gotten sick from them. If you have AIDS, you can actually catch these diseases because your immune system is so weak it can't fight the viruses and bacteria that are just in the air around us. So, but how does this happen? Well, with HIV, the human immunodeficiency virus attacks or destroys white blood cells, which is your immune system. This, the word that they like to use, they say it compromises the immune system. And if you want to be a bit more specific, you can say it's more susceptible to other kinds of diseases if you want, but this is basically all you need to know for that. So again, HIV is the actual communicable disease, so to speak, and AIDS is actually a symptom of that. The human eye, very, very complicated, and as a result, a lot of students kind of hate it because it's a bit difficult. I don't blame you. But we are shown two light receptor cells, so we have a rod and we have a cone that do have two different purposes. Which part of the eye contains light receptor cells? I would probably use a bit of the good old-fashioned process of elimination. So for example, the iris is kind of the coloured part of your eye. It's not going to be that. It's not going to be the lens because that's where the light initially enters your eye. Cornea and retina are both ones that people may choose, but the cornea is actually further forward. All of your light receptor cells are at the back of your eye. It's the retina, which is also where your lens tries to focus all of the light from that enters your eye. So the lens will focus the beams of light onto the retina so that way you can see using your rods and cone cells at the back of your eye. These cells require energy. The organelles labelled X release energy during respiration, so you don't even need to really look at them. You know what they are. They are mitochondria. I almost forgot how to spell mitochondria for a second there. The organelle labelled Y contains chromosomes. Again, you don't even need to look at this really, but if you do, it does give you a bit of a hint. What does that kind of look like? It looks like the nucleus. Then lastly, you have cell A responds to dim light and is responsible for night vision. Name the cell. Well, honestly, it looks like its name. It's a rod cell. Not bad, right? 4 marks in about 30 seconds. Pretty good, but it is a lot of kind of fact-checking exercises. But use all the information they give you. Releases energy during respiration. If you can't remember, try and have a look and see what it looks like. That can help. Honestly, that would help more with the nucleus than it would with the mitochondria. Now, finally, the question that was kind of inevitable is, what's the difference between cell B and A? What's the role? Well, cell A, as we wrote back here, its response to dim light and night vision. If you've ever wondered why your night vision is never in colour, it's because cell A, the rod cells, deal with black and white vision only. Which is why, at night time, you don't see colour. Realistically, you should be able to see colour. But anyways, that's another story for another time. So what's the other type of light? We have black and white vision. We also have colour vision. So cell B is responsible for colour vision. So I would just give its name. It is a cone cell. And it's responsible for, in fact, let's write it in a semi-full sentence, responsible for colour vision. That's pretty much it. If you want, you can say, if you really want to say the opposite of this, it says this is dim light. You could say it's responsible for bright light or intense light, and you get the mark. So that's even nicer. Or you can say something along the lines of it responds to different wavelengths of light, that kind of thing as well. But if you do want to just state the exact opposite, which makes sense, if you have two cells that do the same thing, but have slightly different roles, generally speaking, they're responsible for the opposite thing. So opening and closing, or light versus dark, that kind of stuff. Another maths question / physics question, all linked together in a big, fat, juicy 3 marks, which is a pretty sizable chunk of this question. So, we have the optic nerve is 47 millimeters in length. The nerve impulses travel at 75 meters per second. Calculate the time it takes to travel the length of the optic nerve. Super, super easy. This is our distance. This is our speed. We're rearranging for time. Now, some of you might be okay with just rearranging if your maths is pretty solid, so distance over speed. However, I'm going to be super nice and I'm going to give you a formula triangle. So let's draw out our formula triangle. I'm going to say speed is just S, D, and T. If we want time, calculate the time it takes, we cover up T and we get distance over speed. That's where I got that equation from. Now, that doesn't mean you do 47 divided by 75. This is in millimeters, whereas this is in meters. So what is 47 millimeters in meters? It is, well, you divide it by 1,000, so you get 0.047 over 75. And let's have a look. It should be very quick because that makes sense, right? Like, how long does it take for you to respond to what you see? A very, very, very short amount of time. Can we give it in standard form? Yeah. Does it say give it? Yeah, you can give it in standard form then, that's fine. So I'm going to give it both in not standard form and in standard form, so 6.27 times 10 to the minus 4 seconds. Or if you want to write that fully out, that would be 0.000. The minus 4 means you're going to have 4 zeros before you start writing your number. Those would both be accepted in this case. I'm not going to lie to you, this is basically just a memory exercise. There's not really a way, I mean, there is a way that you can work it out if you know what some of the other functions of the brain are, like the hypothalamus, like the regulatory center, the medulla, maybe. But yeah, it's a bit tricky. But if you know your structure of the brain, this shouldn't be too bad. So the impulse travels to the occipital lobe of the brain at the back. Which part of the brain? It's just going to be the cerebral hemispheres. Not really much of a process of elimination that you can use, unless you know what the other functions are or where the other parts are. Anyways, state the sense most likely to be affected if the occipital lobe is damaged. This is a pretty interesting question because if you're logical, you can work this out. It tells you that the optical nerve, so your eye, will send these impulses to the occipital lobe of the brain. So what sense is affected if the occipital lobe is damaged? Vision? Kind of obvious, right? But you know what? Take it as it comes. Take it as it comes. In 2017, a new strain of Klebsiella pneumoniae bacteria was discovered, which was resistant to 26 different antibiotics. I'm assuming this causes pneumonia, but I actually don't know this disease specifically. Explain how this happens. It's a 4 marker, and I mean, yeah, it's just a 4 marker. So how is it done normally? Well, there's a bunch of things you could say, but the idea is that, when you take antibiotics, the only bacteria that survive are the ones resistant to the antibiotics, which will then go on to replicate and divide. So I'm probably just going to write that. So antibiotics kill non-resistant strains. So only resistant strains survive. By the way, I want to point out that what I'm giving you is a very concise answer. There are other things you can say. So it doesn't mean if you don't write this exact thing you get 0 or you get 3 or whatever. I'll read out some other points that you can get. But again, the way you write it is really important. So if you wrote the same, like, for example, if you wrote something similar to this but not the exact same, you can still get marks. Don't worry too much about that. Only resistant strains survive. Resistant strains go on to reproduce. And remember that they reproduce asexually, which means all of the offspring are clones, which means the offspring inherit the resistance. And if you really want, if you want to be super, super in-depth, when you're studying especially, is when you've written this out, imagine that you're explaining this to someone that doesn't know what antibiotic is. What antibiotic-resistant bacteria are. Would this make sense? Well, okay, antibiotics will kill the non-resistant strains. Only the resistant strains survive. Those strains reproduce and therefore they inherit it. The one thing I might add if I was explaining this to someone is that I would say that random mutations can cause resistant strains. Something like that. But you can also say that it's by natural selection or evolution. You can mention that there's a mutation, like I just said. Some people don't finish the course of antibiotics, something along those lines. Anything else that I can think of? Not really. You can mention that they, like the resistant bacteria, have like a special gene that allows them to survive, which is kind of the same thing what we said. State how the use of antibiotics could contribute to this bacteria developing resistance. Well, when you use antibiotics, if you do not complete the course, or it's overused, then it leads to antibiotic resistance. So you can say people not completing the course. Course of antibiotics. Or you can say overuse. So every time we use antibiotics, we're increasing the number of antibiotic resistant bacteria. Every time. Well, virtually every time. So if we're giving it to millions of people, that's millions and millions of bacteria that are surviving and becoming resistant. So we need to use them. Essentially, you only want to use them if the person's going to die or going to get really, really ill from it. If it's a relatively healthy young person, like I'm hoping all of you are, then you don't actually need antibiotics in a lot of cases. Like a chest infection, you can survive that without antibiotics most of the time. Again, obviously there are people with different conditions. So it's up to your doctor really. But if you do get given antibiotics, it's really important that you complete them. So sometimes you might get 2 weeks of antibiotics, but after a few days you feel better. Finish the course. Even if it means taking medicine for 2 weeks that you don't need, finish it. For the good of humanity anyway. Prokaryotic cell, which is a characteristic feature of it. It has chloroplasts. That's not a feature because that's only in plant cells. It does not have a nucleus, does not have ribosomes. Every cell has ribosomes. It cannot reproduce to that host. Not true, it's a bacteria. So it is indeed the not having a nucleus part. This is a bit of an interesting question because it's only 3 marks, and technically speaking there's about 6 stages. But the idea is, what stages do antibiotic development, what stages happen after we kind of discover a new antibiotic, what stages happen? And it's actually true for every single medical treatment ever. I might actually type this one to save my hands. But the idea is you just need to name the stages. So the antibiotic goes through a development stage. Let's call it a phase, actually. Actually, they use stage. Let's use stage. So this is where we develop or create or invent the new antibiotic. What happens next? Then there is a preclinical stage or trials, which is where we test it on things like individual cells. Or even before that, they may just test it in general about its stability. Then you have testing on animals. So again, the preclinical, maybe you're doing on cells, now you're doing on animals. Then you're going to have actual clinical trials, which when you're testing it on, it depends on the type of treatment, but anyways. Then you may test on healthy volunteers. A lot of people get confused about this stage because why would you test on healthy people? The idea is if you test it on a sick person and it makes them even worse, they're more likely to die. So if you test on healthy people, if there's any side effects, it's less likely to kill them. Simply put, then you would have something like a double-blind trial. As far as I can tell from looking at all of the mark schemes from the previous however many years, you can say just any 3 of these things. Cool, right? You just need any 3 of these and you're fine, and maybe a bit of a description, but that's it. Pretty cool. So he investigated the fat content of 2 types of milk, A and B. He added a drop of oil from a pipette into the test tube of water, and in water it floated to the top. The student placed a drop of milk A into one of water and a milk of B into a different test tube of water. The drop of milk A sank to the bottom, whereas in B it rose to the surface. Why? Well, they actually give you the answer in this question here. The drop of oil rose to the surface of the water. Now, this is because fat and oil float in water. So in this case, the reason why B rose to the surface is because it has a higher higher fat or oil content. So you can just say B has a higher or high fat content. That's it. Which is why they told you that initial sentence in the first place. This is another fun question, and it really does depend on how many experiments you've done in school. So we have 5 cubic centimeters of B and 1 cubic centimeter of lipase were added to a different test tube. The pH of the mixture was initially 7. After it's placed in a water bath for 10 minutes, so you're controlling the temperature, it goes down, it becomes acidic from 7 to H. Why? Well, you need to think about what lipase does. Again, think about what they've included. They've included lipase, but what does lipase do? Well, lipase breaks down fats or lipids, whatever you want to call them. Okay, and what would happen then? Well, what does the breakdown of fats produce? Well, it forms or produces fatty acids, right? And a glycerol. You only need to say fatty acids. Well, fatty acids are obviously acidic. These lower the pH of the mixture. And that's all from, even if I've never done this experiment before, I can work out what's going on by thinking about lipase. What does lipase do? Breaks down fats. Okay, what does that produce? Fatty acids. And the pH has become acidic. Put 2 and 2 together. The production of fatty acids must have lowered the pH of the mixture. This procedure was repeated with milk A. There was no change in the pH. Why? Well, if we go back to question 1, part A, sorry, B has a high fat content, hence it floated. But if you notice, milk A sank to the bottom, which means it has little to no fats inside of it. Which means there's no lipase or fatty acids to break down, right? So simply put, milk A contains little or no fats. Therefore, no or little fatty acids produced. Which means the pH didn't change, because there's nothing making it acidic. The student repeated the procedure with lipase that had been boiled and left to cool, and it was added to milk B. Why did the pH not change? Well, think about it. There's two reasons why the pH wouldn't change. Either one, there's no fats present, hence no breakdown into fatty acids. But this is milk B. We know there's fat. So what else could it be? Well, it means that the lipase isn't doing its job. Why? Can you see my logic here? The process of elimination, the process going on in my brain. I'm thinking of all the possibilities, crossing off the ones that can't be true. The only solution is that the lipase, for some reason, didn't break down any fats. Well, what's the only difference? The only difference is that the lipase has been boiled. Well, what happens at extreme temperatures? Enzymes denature or break down. So all you have to do is write temperature when boiled was above the optimum temperature. Okay? Meaning the lipase denatured, broke down. Which means no fats were broken down into fatty acids. Hence there's no change in the pH. Again, I'm hoping that you can kind of see my thought process. The GCSE in biology specifically, it's changed over the years. Before, it was really just memorizing. Now, there is still memorizing, but you have to also apply your knowledge, and which I do prefer, although it does make it a bit harder. But it does mean you are better at biology. It means you're a better scientist if you can do it. Think about what the possibilities are, eliminate them, think about what's changed. That's the kind of questions you should be asking yourself. This right here is another kind of experimental process that you may or may not have done before. So I'm going to write out all of the steps. There are 6 steps. You only need 4. So I'm going to write all 6 though, just to help you out. So first of all is you're going to use the root tip or the meristem, because that is where mitosis takes place. That's where the growth takes place. So you're going to use the root tip or meristem, whatever you want to use. But you need to choose a place where there is active cell division, which are in those 2 places. The first thing you do is you would soften the root with alcohol or heat or acid or some way to kind of break down those strong kind of cellulose bonds that you have. Then you're going to crush the root. That's why you soften it first, because crushing can be a bit tricky. Technically, you can skip the step if you want. Onto microscope slide. This step is really important, so I'd recommend always including it, but you need to stain the root. You can also name the stain that you're going to use, or like a dye or something. Then you're going to add a drop of water to slide, and then add cover slip. In my opinion, what 2 could you do without? The softening, and maybe even the using the root tip. So these 4 are really, really important in my opinion. Yeah, definitely the staining and definitely the cover slip. But yeah, that's it. Describe what is produced when a single cell divides by mitosis. Remember, meiosis, back earlier in the paper, produced genetically diverse gametes. Mitosis produces clones, identical cells. So what would you say? Well, it produces 2 cells. Meiosis doesn't produce 2 cells, if you remember. It produces 4. 2 cells that are genetically identical. And generally speaking, we're producing body cells. You could also say that it produces diploid cells, which basically means that they have the same number of chromosomes as the parent. I would say this is probably the weakest point, the producing body cells. The most important is that it's 2 cells that are genetically identical with the same number of chromosomes as the parent. Which is important again because in meiosis it produces half the number of chromosomes in the parent, which is why they produce 4 cells, by the way. Hopefully that makes sense. Student observed 89 cells on the microscope slide. These are each stage of the cell cycle, prophase, prometaphase, anyways. use this equation to calculate the mitotic index. So the number of cells that are actually doing mitosis over the total number of cells. So it's not going to be 89 over 89. Which of these phases actually have mitosis happening? It's all of them that are not interphase. Interphase is this kind of like in-between phase between stages of cell division. So what you're going to do is add up these three numbers. So 12 plus 16 is plus 6 is 18, plus 18 is 36, plus that is 45. So you're going to do 45 over 89 times 100, and you're going to get an answer of 50.6%. Half of them were in mitosis. For 3 marks, that's pretty good. Oh, I sneezed right after I said that. That's why I ended that last clip really abruptly. Wow. The mitotic index is often used in the diagnosis of cancer. State the effect of cancer on cell division. Cancer, in general, is literally just uncontrolled cell division. So it makes cell division uncontrolled. So these phases will just happen indefinitely and happen a lot faster than usual. Scary thought, but that's all it is. The theory of evolution and natural selection is, it's actually often, again, misinterpreted, because what some people say is that there's a selection pressure and then there's an evolution of the species, which isn't true. In general, it's because members of a species have variation in their genetics. A selection pressure comes along and will kill, or only the best adapted will survive, or the best adapted are more likely to reproduce. And so over time, you see that genetic trait more present in further species, which is also why we still have, there are certain members of a species that are better adapted than others of the same species. But that's simply because they're more likely to reproduce. So it doesn't just wipe out all of them that aren't adapted. Or, it's like one of the common things is the idea that if the world floods, human beings would evolve to have gills. No, they wouldn't. It's just that the people that are best adapted to living in the water will survive and reproduce. That's all it is. We wouldn't just spontaneously grow gills. But anyways, that's enough of me being weird. I'm going to give you 6 points. You only need 3 of them. Actually, I'm going to give you 5 points. So, really core idea is that organisms in a species have mutations or variations. That is a core concept of natural selection or the theory of evolution. It's a core concept. Then, a new selection pressure emerges. So it could be that the temperature of the globe changes, the amount of prey changes, the amount of food changes, whatever it may be, the amount of space, a new predator, whatever. And organisms that are best adapted to the pressure survive and reproduce more than those that are worse adapted. You do not need to write that entire thing. You can just say organisms that are adapted to the pressure survive and reproduce. I've added this last bit because it really annoys me that the mark scheme doesn't mention it, because it gives the wrong idea. Even members that are not adapted can survive and reproduce. Of course. Again, that's just something that annoys me a bit, but whatever. So the offspring inherit the adapted trait. And then over time, this trait becomes more prevalent. God, I knew I spelled it right the first time. No, wait, what? It looks wrong both ways I wrote it. Anyways, becomes more prevalent. That means that more members of the species will have it. But again, it doesn't mean all of them will have it. But anyways, story for another time. So that would be, you only need 3 of those marks, but hopefully that gives you a good summary of what natural selection actually is, okay? Oh, I just realised we're adapted. Alright, cool. So these 2 gentlemen did not always agree. So Darwin believed that male birds have feathers that are brightly coloured to make them more attractive to female birds, whereas Wallace thought that female birds have less brightly coloured feathers so they're more likely to survive. they're basically asking you, why would that make them more likely to survive? Well, simply put, it makes them less noticeable. So, less brightly coloured feathers are less noticeable, which means you are less likely to be eaten or killed by predators. That's it. And that's it for that question, right? Anything else you can say? Well, actually not. You can also say it's less likely to be seen by prey. So if you imagine, if you're trying to sneak up on your friend and you're wearing a brightly coloured, flashing lights and all that stuff, you're more likely to be spotted. Whereas if you're camouflaged, you're more likely to actually be able to like jump up on them. That kind of idea would also work. So just why it's more important that the female birds survive more frequently than male birds. And that's actually, again, what they were saying back here, the idea that the male birds have brightly coloured feathers, which means they're more likely to be eaten and killed. The female birds have less brightly coloured feathers because they need to survive. But why is that? Well, the idea is a few things. Is that female birds are the ones who lay the eggs. Or produce the offspring. And here's the big part, is that one male bird can reproduce with many female birds. Imagine if you had 10 males and 1 female. That female can only produce a certain number of offspring because they only lay a certain number of eggs. Whereas if you had 1 male and 10 females, that 1 male can have kids with all 10 of the female birds. So that's the kind of idea that why it's more important for females to survive than males. This is probably one of the hardest biology questions I've come across. So we have the limbs of 5 animals. We've got a human, cow, horse, whale, and a bird. How the structure of these limbs provides scientists with evidence for evolution. There's a really fancy term you can use. I'm going to avoid it, but I'll tell you what it is afterwards. You can look at this and see that there are similarities, right? So I'm going to say that there is these 5 animals share a limb structure. Right? Can you see how they look? They kind of all have a similar limb structure. This looks like a hand, looks like a hand, looks like a hand, looks like a hand. This terminology is called a pentadactyl limb structure. Penta being 5, because if you notice there's 5, 5, 5, 5. And so pentadactyl limb structure means that there's 5 digits at the end of the limb. But anyways, they share a limb structure which suggests a common ancestor. Right? There's an original that had this kind of limb structure that then differentiated into multiple different species. And in these 5 animals, the limb structure has adapted to different purposes. So for example, if you look at the human one, look how many different bones there are here. That's because we use our hands for gripping things and holding things and manipulating things. Whereas a bird, they have giant long fingers, in air quotes, because it's used for wingspan, it's for flying. They don't need to grab things with their wings. Same with the whale's fins, although you do have more bones here. That's because with whales, when they swim, you need to have more motion in the inner fluid. And then obviously a horse and a cow is more like a foot structure instead. Pretty tricky question, I'm not going to lie. Pretty tricky question. If you mention the pentadactyl limb, that actually gets you a bonus mark with this limb structure part. But I don't really know of many students that know that. But if you do, well done. I really was interested in this question back when I did GCSEs. This was actually a type of question they used. They do use it sometimes, but it's not super successful. But anyways, there's a shortage of kidneys and every organ. For organ transplants, they're investigating how to grow kidneys in pig embryos. Interesting. So they take human stem cells from the patient, they put it into the early stages of a pig embryo, which will then reproduce to produce human kidneys rather than pig kidneys, and they're put straight back into the human. And the benefit for this is that they are genetically coded to you. For all intents and purposes, these kidneys are your kidneys, which means, and the big problem with organ transplants between humans is that there's often your immune system will reject someone else's kidneys because it identifies it as a foreign species, so to speak, and attacks it. So actual human kidneys. Cool. So why does it have to be engineered so it doesn't grow pig kidneys? And might be an obvious reason. It's because it would be rejected by humans. So pig kidneys rejected by humans. Pig kidney reject by humans. Right? The idea is you do need, in an ideal world, the organ transplant you get has to be genetically identical to you. So that way your body doesn't know any better. So why human stem cells are used for this process? Well, what are stem cells? Stem cells are undifferentiated cells. So they can produce the kidney cells. And if you want, that won't get rejected. Simply put, right? Again, the whole idea behind this whole process, this lengthy process, is that you have genetically identical cells in your body that your immune system doesn't try and reject and fight back. Probably one of the most depressing graphs I've ever seen. So the number of organ transplants needed has skyrocketed. This is because people are living longer, mainly. And so obviously when you get old, your organs run out, they start dying. But the number of donors has stayed pretty much the same, which sucks. So compare the number of donors available with the number of organ transplants needed between 1991 and 2018. Well, you could simply say that the first one is that the first thing is the number of transplants has increased rapidly, but the number of donors is approximately the same. That's one good one. You can also say the number of donors, because we need a third mark, is always less than the number of transplants. And that will give you your 3 marks. It's literally just describing the graph. You can also say that after 2000 and... Actually, yeah. I mean, no, you can't because it's up to 2018 only. So after like a certain point, it does dip a little bit, starts to decrease. That is also a valid point you can make as well. So why are scientists genetically engineering animals for organ transplants? Well, simply put, there's not enough donors. So there is a demand for it, right? We need this many organs. We only have this many available. So we're going to try and genetically engineer animals to bridge the gap. So that way we can, treat everyone. For part C, there is a little asterisk here, right? And that means that they're marking or grading your quality of written communication. Now, this does not mean you have to write in full sentences. It doesn't. It means that when you present the steps, they are given in a logical order. That's pretty much it, and you don't contradict yourself. So, for example, you would start off with... Where you start off with isn't too particular, but you need to make sure that everything that comes afterwards is actually afterwards in the process. So, for example, the gene that codes for human insulin is identified in DNA. Then it's removed using restriction enzyme. And a plasmid removed from bacterial cell. You can say what you're using lysosome if you want. Plasmid cut open, and using the restriction enzyme leaves the complementary ends open. Human gene inserted into bacterial plasmid. Plasmid returned to bacterial cell. Bacterial cell multiplies. And the idea here is that it then continues to produce insulin. I have actually, believe it or not, I've actually trimmed the answer down. So for example, I didn't mention the lysosome, and I didn't mention some other things as well, like using the same restriction enzyme to cut open the plasmid, because I wanted to limit it so you can still get the 6 marks without writing, an entire page essay on it. Fiatencio is part of a method used to produce a bacterial culture on a petri dish, and they're talking about why step 1 and 2 are necessary. So sterilizing the dish in agar and passing the inoculating loop through a flame. It's simply to prevent contamination. Contamination. So the idea is that if you have viruses or bacteria already on either the agar or the loop, then you're not going to get the exact bacteria that you want in this case. So yeah, killing microorganisms, preventing other microorganisms affecting results, etc. Allow inoculating loop to cool. Why is that included? Simply put, so the bacteria are not killed. Because if you put a boiling piece of metal in bacteria, it will just kill the bacteria instead of allowing it to actually be transferred. The student wants to investigate how effective 3 different antiseptics are or were at killing bacteria. They're provided with this petri dish, different antiseptics, filter paper discs, and sticky tape. So devise a plan. Now, I cannot actually give you a mark scheme answer. I mean, obviously there is a mark scheme, but there are a few different methods, and so I'm going to talk through the logic behind this. Is you're testing 3 different antiseptics. That is your independent variable, because that's the thing you're changing. The dependent variable is going to be how much bacteria is on the agar jelly. So what you're going to probably do is measure the radius or the zone of, what do you call it? A zone of inhibition. So the area where there is no bacteria. The size of that area will tell you how good of an antiseptic it is. So the smaller the area, the worse the antiseptic is. So if you're trying to find the best one, how effective it is, you're looking for the one with the biggest radius. And you measure that with a ruler, for example, or you can use a camera with a ruler in order to work it out. Using pi r squared to work out the area. What kind of safety measures will you have? You can use things like work near a Bunsen burner, only lift the lid slightly when you're talking about the bacteria, because otherwise breathing it in. The sticky tape is used to stick down the lid so there's no spread of bacteria. Washing your hands. What kind of control variables will you have? Well, you want to incubate them for the same amount of time. So I would prepare all 3 of these dishes at the same time and leave them for, let's say, 2 days, and then check. If you do it one at a time, you run the risk of doing it in different conditions, so different temperatures, things like that, and also for different lengths of time. You want to use the same size filter paper. You want to use the same temperature. So again, doing it all at the same time and putting them in the same place is a good idea. The same amount of antiseptic as well. And yeah, you also want to label the petri dish, things like that. But again, in terms of what you want to do is put the different antiseptics on the filter paper, on the agar jelly, leave them in like a cupboard for 2-3 days, take them out and measure the radius of the uncontaminated area. Then you get to see which one's biggest, and that's the idea. But the sticky tape is used as like a safety measure. Lastly, but not least, viruses can cause disease. Describe how the lytic pathway is involved in the reproduction of viruses. Okay, so what happens? Well, the virus will insert its genetic material into a cell, into a host cell, technically. The host cell's organelles will reproduce the virus, basically. The virus components are assembled in the cell. And the cell lyses. So what is lysis? It means it ruptures, it splits, releasing the viruses into the bloodstream. That's it. So essentially the lytic pathway is the method by which the virus will insert its genetic material into the host cell, eventually causing it to rupture by filling up the viruses. And that's essentially how it works. But that's it, 100 bloody marks. So in theory, you should only have an hour and 40 minutes to do this paper. They've given you an hour and 45. A whole extra 5 minutes. How nice of them. Pretty hefty paper. On my Discord, I have, don't tell anyone, but on my Discord, I have put the 2023 paper 1 and mark scheme in there if you want to have a look under the announcements tab, just to help you prepare. I'm not allowed to record it. At least in theory, I'm not allowed to record it anyway, so that's why I haven't done that. But anyways, best of luck on Friday, and I'll see you in the next one.